In 2018, I traveled to England to research the mysterious rare disease that I've had since birth. My first stop, Queen Square, London. This is where the story of Wilson disease began. It was here, at the National Hospital for Neurology, that Samuel Alexander Kinnear Wilson made a landmark discovery. In 1912, the American-born British-trained physician identified an illness with an unlikely combination of characteristics: changes in the brain and cirrhosis of the liver. The disease was always fatal in the young patients he observed.
He called it progressive lenticular degeneration.
Dr. Wilson's son, James Kinnear Wilson, lives nearby in Cambridge and remembers his father's discovery, and when hepato, or liver, was added to the name.
It's been called hepatolenticular disease. But I don't think he would have liked that, because hepas is Greek, and lenticularis is Latin, and he was a classical scholar, and I'm sure he would have said they don't mix.
After Dr. Wilson published his finding, it was given a new name, Wilson disease.
I think he was very pleased about it. Very proud of him. Yes, it's quite an honour. He presented the thesis to Edinburgh University and received a gold medal as a result of it.
Despite Dr. Wilson's continuing research, discovering the cause of the disease eluded him.
He didn't know what caused it. He got as far as saying that it was toxic in origin.
Dr. Wilson was right. Doctors discovered decades later that a genetic defect allows copper, an essential dietary mineral, to accumulate in the liver and brain, causing a variety of devastating symptoms, as can be seen in this film from the 1980s.
This liver disease may cause swelling of the abdomen, jaundice, bleeding, as relentlessly worsening tremors or rigidity of the hands, feet, and head, drooling and slurring of speech, and abrupt personality change, the sudden appearance of impulsive, grossly inappropriate behavior, often sexual in nature, temper tantrums, inexplicable deterioration of schoolwork, or the appearance of a psychosis that cannot be distinguished from schizophrenia or manic depression.
Everybody with a diagnosis of Wilson's disease is different. And from what I've seen of the Wilson disease patients, it is absolutely true. They're all different.
In my case, Wilson disease put me in liver failure when I was a college student. I was lucky. I was diagnosed. By then there was a treatment, and it worked. I've had a normal life and spent my career reporting medical news. I've been living with a diagnosis for 35 years and trying to understand it better. I found out that Dr. John Walsh discovered the first treatment for Wilson disease. He's 98 years old and also lives in Cambridge. So when was the first time you heard about Wilson disease?
I had a Fulbright fellowship to the States, 54, 55. I was working with Charlie Davidson, who was a liver doctor. And we were asked to see a Wilson disease patient who'd gone into liver failure.
They couldn't help the patient then, but crossing the bridge from Boston City Hospital back to Thorndike Laboratory, Dr. Walsh had what he calls an inspiration.
I said to Charlie Davidson, you know, what this chap really wants is penicillamine. And Charlie Davidson said, what's that? I discovered this new amino acid which had never been seen in human urine before.
Dr. Walsh had previously discovered penicillamine in London during the early 1950s while studying lab samples from people who were given the antibiotic penicillin.
And then it was just put in the memory bank as an interesting observation of no particular importance.
To prove his theory, a chemist from MIT then provided Dr. Walsh with some penicillamine, and then he did what would be unheard of today. He tried it on himself first.
But it didn't do me any harm, and the next day I was alive and well, so I decided it was safe for me, it was safe for the patient.
So what happened then when you tried it on the patients in Boston?
It got copper out.
Soon after, Dr. Walsh's Boston fellowship was over, so he returned to London with a small supply of penicillamine and tried it on some patients.
My father was Britain's leading neurologist at the time, and he found some Wilson disease patients for me to try it out on.
One of them was Shirley Wiley. It's a medical marvel that she can draw this spiral. In 1955, she was facing almost certain death until a series of remarkable coincidences brought Dr. John Walsh to her bedside. What did she look like when you first met Shirley?
A mess. She was bedridden and helpless.
Shirley was 15 when she was taken to a London hospital because no one could help her.
If he hadn't come over here, I wouldn't have been here.
And how long did it take to start seeing her get better?
She was better at the end of a year.
Her symptoms had reversed, and in 1956, Dr. Walsh reported in the American Journal of Medicine that his drug discovery worked. Do you remember how you felt when you started to get better?
Yeah, I was walking, I was doing everything.
How did that feel to be able to walk again?
It felt lovely.
Dr. Walsh continued his research in Cambridge and found that Wilson disease needs to be treated every day for life. The results of his drug discovery were dramatic, but he also found that penicillamine does not work for everyone.
We ran into trouble with penicillamine. I wanted an alternative treatment, and my laboratory was literally a couple of paces away from the university laboratory of biochemistry. And one morning I wandered over to the University of Biochemistry, ran into a biochemist called Hal Dixon. I explained my problem to him, and he took a bottle off the shelf. I said, triethylene tetramine, very similar to sparadine, should be non-toxic, is known to bind copper. Try it. And that's how we got tetratriantine. So it was Hal Dixon's idea, and my work proving that it worked and was safe.
Do you feel like one is superior to the other?
I think it's a question of which one works on best on which patient. I think it's a question of trial and error.
Penicillamine and triantine turned Wilson disease from an always fatal disease to one that's treatable. Since then, researchers discovered that zinc works as a third treatment option. Shirley's taken penicillamine for 63 years. So what do you want other people with Wilson disease to know?
Oh, to keep taking the tablets. Please don't stop.
She's been able to live a normal life. She married, and while raising her 3 children, she worked at Scotland Yard cleaning offices. You were a medical pioneer trying penicillamine.
But there's a lot of people better by taking it, isn't there?
How does that make you feel?
Proud, happy.
Dr. Walsh asked his patients to draw spirals to monitor their progress. Shirley keeps this London Times article that told the story of her remarkable recovery as a reminder of how far she's come since she drew her first spiral in 1955. What do you think when you think about Dr. Walsh?
It's marvellous. I wouldn't have been here but for him.
Well, I think it is a remarkable series of coincidences that all worked out well. The coincidence that I discovered penicillamine, which was a coincidental discovery, the discovery that we saw patients with Wilson disease, and I realized that penicillamine might work, and the coincidence that I was able to get some, and that it was able to show that it did work. And the whole thing was, if all those coincidences hadn't worked out, the whole idea might have gone fat.
It's incredible.
Yeah. It was all good luck, really.
Walking across that bridge and having that idea, I mean, where did that come from?
Where did it come from? Someone up there. Where do ideas come from? Where do ideas come from?
But the story doesn't end there. For too many people, getting the diagnosis and the life-saving medication in time, or at all, remains the challenge of Wilson disease.
Hello and welcome to today's webinar on gene therapy. I'm Rhonda Rowland and I'll be your host and moderator. Today we have a lot of firsts to talk about. If you were able to join us earlier and watch the story of Wilson disease, you know the first treatment was identified by Dr. John Walsh almost 70 years ago. Today's webinar is a first for the Wilson Disease Association, and it's the first one that I've hosted, and I'm truly honored. I'm a Wilson disease patient myself, a member of the WDA's board of directors, and my former career was as a CNN medical correspondent. While at CNN, I reported on the Human Genome Project. As part of that medical milestone, the gene for Wilson disease was identified in 1993. Now, almost 40 years later, we have another first. We have not one, but 2 gene therapies that are currently being studied. I'll be joined by Dr. Fred Askari, who leads the Center of Excellence for Wilson Disease at the University of Michigan, to talk more about these gene therapies. But first, we'll watch this excellent but short video produced by the American Society for Gene and Cell Therapy, which explains how gene therapy might help people with Wilson disease.
Wilson disease is a rare genetic disorder that causes excess copper to be stored in the body. The disease is progressive, and if left untreated, can cause liver disease, brain damage, and premature death. To understand Wilson disease, it's important to first know the role that copper has in helping the body function. Copper is an essential element that supports the nervous system and immune system and helps make blood cells. Typically, copper is absorbed from the foods we eat. The ATP7B gene provides instructions to produce a transporter protein that moves excess copper out of the liver. Wilson disease is caused by mutations or changes to the ATP7B gene. The transporter protein doesn't function properly, meaning its activity level is low or absent. So excess copper stays in the liver. It builds up and becomes toxic throughout the body. Symptoms typically begin due to liver dysfunction during childhood or teenage years. Signs of advanced liver dysfunction can include chronic fatigue and yellow discoloration of the skin. And a small number of people with the disease will experience severe liver failure. But in some cases, liver disease may progress silently, while patients initially develop symptoms from the effects on the brain, such as tremors and difficulty speaking or moving. For some, the effects of Wilson disease can be prevented or treated by lifelong medical treatments that are often given daily to lower copper in the body to non-toxic levels. However, these available treatments aren't effective for everybody, and that's where gene therapy may be able to help. Here to explain how gene therapy can treat Wilson disease is Dr. Michael Shilsky.
Gene therapy aims to be a one-time treatment that delivers a working ATP7B gene into cells that produce working transporter proteins. The goal is to restore proper liver function to remove excess copper. An in vivo gene therapy approach is used, meaning it's delivered directly into the body. This is done via intravenous infusion to target liver cells.
Scientists know that viruses are good at getting into cells, so they have learned how to safely use this ability to get into cells as a vector or carrier that delivers the working gene. But don't worry, the viral genes are removed, so only therapeutic genes are delivered. Think of a viral vector as an envelope with a message inside. The harmful letter that could cause disease is taken out of the envelope and replaced with a new gene that has a specific message. Once delivered, the working gene gives instructions to liver cells to make the transporter protein that helps the cells remove excess copper from the body. Doing so aims to prevent, slow, or stop disease progression. But it is unknown whether this treatment can reverse the existing damage. This investigative therapy is currently being researched in clinical trials. Clinical trials are part of a required research process to determine if a treatment is safe and effective. Researchers are optimistic that gene therapy may be an option for some individuals affected by Wilson disease. For more information and resources about gene therapy for Wilson disease, visit patienteducation.asgct.org.
Well, as promised, we're now joined by Dr. Fred Askari. Dr. Askari is a specialist in hepatology or liver disease, as well as in medical genetics and liver transplantation. Aside from his expertise, we were also interested in interviewing him because both gene therapy trials are being studied at the University of Michigan. Dr. Askari is principal investigator for one and a study site for another. So I'm just now waiting for Dr. Askari to join me on camera so we can start asking him the questions. And there he is. Hello, welcome. Thank you for joining us.
Hi, thanks for having me, Rhonda. I'm looking forward to this.
Well, as I just mentioned, you are the principal investigator for one of these gene therapy trials and a study site for the other. Could you explain what that means?
Well, principal investigator is really a term we use more in academic context. I'm really the coordinating investigator for the ultragenic study. And the coordinating investigator role really is to sign off, or they do this trial at many centers across the world, and is just to sign off on the regulatory documents, be sure that everything is in order from the perspective of the participating investigators. So it's really more of a regulatory role in terms of what's being done, rather than what you're calling a principal investigator. I wouldn't refer to myself that way. So, but it's we're the coordinating. I'm the coordinating investigator for that. I was very pleased to be picked to do that. And then the site,
study location.
The study location, obviously there are many study locations for both of these trials. And what that means is that we're one of the study locations for the Vivette gene therapy trial. And obviously I've worked very closely with Vivette as well, getting the study up and open to enrollment at our site.
So as I mentioned earlier, with the first treatment that was identified for Wilson's disease, Dr. John Walsh actually tried the drug on himself first, which would be considered completely unorthodox in today's standards. And because he was fine the next day, he went and tried it on patients. Can you explain just how the scientific process has evolved from that time to ensure that these treatments are safe and effective for patients?
Well, there's, the FDA is a lot more involved in clinical trials and studying drugs than perhaps they were back then. People have to file for IND before using, which is investigational new drug. There's been a lot of background work done and looking at, is this is a treatment safe, effective? What are the potential toxicities? What studies have been done in animals or in other diseases with similar drugs? What's the human toxicity data that's available before a drug is introduced into a clinical trial? And so someone has to file for this IND. In this case, the companies have filed for these INDs for these two gene therapy products and have shown a bunch of data to the FDA and since these are biologic therapies, to other committees as well that have reviewed it. And then we get permissions to do it. People review what patients are being told in the informed consent documents. And those are approved by institutional review boards. And only then can someone commence doing a clinical trial the way that was so casually done in the past on a I'll take a pill and see if it's okay for me and then give it to somebody else. So that's something of the past.
So a lot of checks and balances today. And from what I've been able to discern, as far as the differences between the Ultragenics and the Vivette Pfizer trial, is really in the study design. Can you talk more about that?
Right. Well, there are some differences between the two trials. They're both using what are called adeno-associated virus vectors. but obviously they're using different producer cell lines and slightly different what we call backbones or plasmids inside of these vectors. And so there are some slight differences there, and then there are also differences in the study design. The study inclusion and exclusion criteria are slightly different. So there may be someone who is eligible for one trial and not another trial. That's just, two groups sat down to write a clinical trial and put down different inclusion and exclusion criteria. So if you're not a candidate for one trial, you could potentially be a candidate for the other one. So that's another difference. And then in terms of the study design, what you pointed out, the biggest difference, I think, is that one is a randomized trial where people are getting upfront in a 2-to-1 ratio, which means you have a 2 times chance of getting the vector up front to a 1-time chance of getting a placebo up front. And then everyone is getting offered vector at the end of 48 weeks who got the placebo versus everyone getting treatment up front. There are also some differences in the monitoring in terms of the number of visits that are being had for each trial, as well as the Vivette study incorporates infusion of a very tiny dose of radioactive copper as a tracer to monitor if the gene therapy is changing the way copper is metabolized. So those, I think, are the biggest differences between the two studies. There's also a slight difference in immunosuppression afterward, where one study is using prednisone after the gene therapy infusion for 58 days, and the other study is also using sirolimus, which is called an mTOR inhibitor, another drug that can have some slightly different side effects. So those are the two. Those are the main differences, at least as I see them. Although there, probably hundreds of subtle ones if you really want to break down the differences between the two studies without going into any further detail.
And I think that it's important to point out that one study is not necessarily better than the other. And in a way, it reminds me of Dr. Francis Collins and J. Craig Venter, who were each working on the mapping of the human genome, and they had their competing approaches, but in the end, we all benefited.
Right. Basically, this is, in my mind, a very healthy competition. The way that these gene therapies are developed actually in the laboratory is a competitive process. The producer cell lines or the candidate gene therapies are all screened very carefully leading up to this, and in the lab to see which ones produce well, which ones express well, which as you're making the vector, there's a screening process that goes on, and it's a competitive process, and it's a meritocracy. The best vector wins in the lab. This is going on at each one of these companies. And we're very fortunate as a Wilson disease community to now have 2 vectors that are similar but slightly different from each other in 2 trials that are similar but slightly different from each other. So that we really will hope to see, how well these vectors work. And my hope is that both of them will work extremely well, and patients will have choices from 2 companies going forward. But we'll see.
And that would be very exciting for sure. And at this point, before continuing, I'd like to mention that Dr. Askari will be taking our audience questions later in our program. So if you have a question, please enter it in the question and answer box on the left side of your screen. So now we'll go on to talk about who would be a good potential candidate for one of these trials. Since we've talked about the studies themselves, who might be an appropriate candidate for inclusion? I mean, are these trials open to just anyone with Wilson disease?
Well, I think the simplest way to answer your question would be that both trials want people who are somewhat stable on their treatment. So what neither trial wants to do is try and catch a falling knife, so to speak, take someone who's deteriorating rapidly and put them in a clinical trial. What we'd like to do is enroll people who are taking a stable dose of copper control medicine, and their copper is fairly well controlled. So I think that's the probably the overarching concern. And then the second concern is that they're erring on the side of enrolling people whose liver disease is not that severe. So if someone has horribly decompensated liver disease or has a lot of problems with liver disease, in general, they wouldn't be a good candidate. And similarly, someone who's psychiatrically unstable, is requiring recurrent psychiatric admissions, or is having lots of psychiatric problems, they probably would be someone that wouldn't be treated in the beginning of this trials. Similarly, neurologically, some neurologic problems are being allowed in the trials, but obviously someone who severely has severe neurological problems would also be not being looked at to be the first people in the trials. So those, I think, are the overarching things. And then there's screening done. We look for antibodies to AAV, the vectors, to be sure that there are no antibodies that'll make the vectors not work. We also look for any pre-existing conditions that might be a problem. Some of the studies have very strict cutoffs, for example, with regard to triglycerides or other parameters that we're looking at. Obviously, any decompensating liver disease would be an issue for either of the trials. So those are the kind of the things that we that we would look at in screening. Active or severe alcoholism would be an issue too. So being willing not to drink during portions of the trials is an issue for some of the trials. So those are, I think, some of the considerations.
So you talked about what you're looking for in the screening process, but from the patient perspective, what's involved as far as time, how they go about it, number of visits, those sorts of things?
Well, I think if you're talking about the screening visits, there's a couple of them for both trials. And basically, if they're interested in participating, the first step is to reach out to one of the study coordinators at one of the sites. And obviously, our site is delighted to screen anyone who wants to be screened. We can, and many other sites are too. And those can be found on clinicaltrials.gov. So I want to be clear, we're not the only site that's screening people. All these sites are, and I think they'll all be happy to screen people. And I would say the first step would be to reach out to the study coordinators at those sites and get screened, and that will help figure out whether they'd be a reasonable candidate or not. I always like to encourage anyone who thinks they might be a candidate or thinks they might, rather than judging for themselves whether they would be a good candidate or not, feel free to reach out, and we'll try and help address those questions that they may have. The screening process itself occurs over a period of weeks, involves coming in, doing some baseline testing. Each trial is slightly different, so they involve slightly different tests, but really assessing the severity of the Wilson disease, the severity of the impairments from Wilson disease, if any, filling out a bunch of questionnaires about their health, about their their current problems or what's bothering them from Wilson disease, if anything, and then also drawing blood work and doing imaging and those kinds of things. There's options to have a liver biopsy if people want to, but they're not required to have one. And then obviously, when the screening is completed, then they can go forward and get enrolled if they meet the eligibility criteria. And both of these studies have very strict review of eligibility criteria. So, this is a safety measure, but they want to be sure people are candidates for this study and are, appropriate candidates. And so basically what that means is that the eligibility is centrally reviewed and signed off on, and then the person can be enrolled if they wish to be. So it is a process, but we're talking about a couple of visits over a period of up to 12 weeks, depending on the study, to complete the eligibility screening.
And, after going through this rigorous screening, there may be people, as you mentioned, who may not be included. And of course, none of us like to be rejected ever, and especially if you're stepping forward to give up your time and you want to contribute and also have the potential benefit. It may be just kind of hard to get that answer. So what would you say if somebody is facing that?
Well, I actually would view it as not a rejection or a no. I would say it's more of a not now, would be a way of looking at it. And we thank everyone for their time and, their participation. I think it's great. And you're actually making a contribution even coming in for the screening to helping us and the companies better understand Wilson disease and better understand what the characteristics are of the people who want to participate. These protocols are also going to be and are constantly being looked at, revised, whatever words you want to say. So if someone isn't a candidate today, that doesn't mean they won't be a candidate to get rescreened in the future and enrolled in the trial. Obviously, the bias is towards safety. I believe these are both very safe trials, but they're being conducted, to screen patients for safety. And as we get more safety data, then the eligibility criteria may be changed. So someone who isn't a candidate today might be a candidate in 6 months or a year or after the drug is approved even. So I'd like to be encouraging in that regard, and I wouldn't view it as a rejection so much as a timing issue.
Well, that is encouraging. And as you mentioned, for more information, you can go online and look at clinicaltrials.gov. And there's a search box there where you enter Wilson disease, and then you can search out these trials and find out more information. And now we'll turn our questions a little bit to dispelling, some of the fears that people might have about gene therapy if they're considering being involved. It's very exciting, certainly, that we have these two trials, and at the same time, just trying something new can be a little scary. You might feel a little anxious. We hear about these viral vectors and having them injected. Exactly what does that mean? Because it can sound kind of scary.
Well, yeah, it can sound scary, but these are basically the coating or the shell of a virus that's being used. It's not the whole virus. So what's happened is producer cell lines have been generated in a lab that make a viral particle, which is the outside coating of the virus, with the genetic information for the Wilson's disease protein, the ATP7B, inside of the viral particle. And so it's not a particle that replicates. It's not an infectious particle where it makes more of the virus in your body once it goes in. It's basically a coating that delivers the genetic information for Wilson disease to the liver and allows the liver now to make normal ATP7B protein or near normal ATP7B protein. And so that's the whole goal of this gene therapy, is to deliver that genetic information. So it's a viral particle. It's not a virus, and that's an important distinction. It's not going to create more virus inside of your body. It's designed to be infused, so it's to a patient who's ever had a bag of IV fluids. It's getting this is very similar to that. It's the vectors are basically reconstituted into a bag of IV fluid, and an IV is started, and the IV vector is run in through a peripheral IV, and it hones in and goes into the liver and delivers the ATP7B gene, which then produces normal ATP or near normal ATP7B protein at that point. So that's what's involved, and why it's quite different than a virus itself.
Okay. And the other very real concern is there have been some deaths from gene therapy in trials for other diseases. What have we learned from those tragic losses?
Well, I think obviously those are our tragic losses. I think we've learned something about dose response in the case of the most recent trial that had losses, and it was actually people were concerned about the doses of vector that were being used. So when we talk about vector dosing, we like to talk about the number of viral particles that are, these particles of genetic information I was talking about that are being infused. And in this trial, they were using much bigger number of viral particles than what's being used in this gene therapy studies that are being proposed today. So the starting dose, for example, in one of the trials, is basically 100-fold less than the dose that was used in this trial where there were deaths. So it would be as if someone were comparing the dose of zinc and saying, well, some people died if they took, if you were on 150 milligrams a day of zinc, and say, well, some people died if they took 1,500 milligrams a day of zinc. So it's 100-fold more in terms of the amount of virus that was being used. So that's one very important distinction, is the amount of viral particles that are being infused. And so we talk about this as the dose response or the therapeutic index of the drug. And basically what that means is as you take more, it gets more toxic. So that's one difference. The other difference are these were different vectors. These this vector was an AAV 8 vector where they had which is adeno-associated virus subtype 8. In one of these trials, they're using an AAV 9. They're not using AAV 8. So these are basically differences that I think are very important potentially. And then finally, the disease being treated is quite different. We're talking about a disease where these children were dying, where they had a muscular dystrophy, and the target was the muscle, and their livers were sick. They were quite sick. So when you put all those things on, I think what we've learned is to be careful about screening who goes into the studies. Be careful about the disease we're treating, what disease is it that's being targeted, and then also be careful with the dosing. And so those considerations are all being taken into account in both of these trials.
So to date, we just have about a handful of diseases where there are proven gene therapies, on the market for them, but we still have had a number of studies. Can you talk about just the sheer number of studies that have taken place and the huge number of other diseases that are now currently being studied as well with gene therapies?
Right. So what we're talking about in terms of experience with this is literally hundreds of diseases and thousands of patients have been studied. So if you look at, for example, one of the companies that's doing this, this is their 5th disease where they've dosed a patient with treatment. They have 4 other diseases that are, have been studied and are on the market, and they have no severe adverse events and no deaths in those studies. So So I think these are viewed. I don't want to minimize the concerns about the people who died in the other trial, but these are viewed with as fairly safe treatments. I mean, we're hoping these will be safe and well tolerated treatments like they have been for other diseases. The big difference here is we're treating Wilson disease, and so that's a new disease. So I think that's the area that's I can't tell you, we know what it'll do in Wilson disease definitively. Otherwise, it wouldn't be a clinical trial. So there has been one patient dosed in one of the studies already, and so there'll be more.
Well, it looks like we've lost Dr. Askari there for a moment until we get him back on. I think one of the exciting things he just mentioned is that in one of the trials, we have already had one of the patients infused with the gene therapy. So that is an exciting milestone. But perhaps while we wait for Dr. Askari to come back online, we do have some polling questions that we have for you, our audience, to find out a bit more about you and what your needs might be or knowledge of gene therapy. So if we're able to get that polling question up on the screen, our first one does ask the question about how long gene therapy has been researched. And this is anonymous. Your answers, nobody will know if you get it right or wrong. You can just click on the screen. The first question or the first option is A, 1 to 5 years, B, 10 to 30 years, C, 30 to 50 years, and D, since 1968. And I'll just give everyone a moment to ponder this. It looks like right now, answer B seems to be winning the race here. I did mention earlier that the gene for Wilson disease was identified in 1993. But I can tell you that the answer is actually D, since 1968. So even though the human genome was being mapped during the 1990s and was actually just fully completed in the year 2020, it was announced in about 2000 that it was completed, but there was still some genes that were not fully identified, and that was due to the fact of technology limitations at the time. So now the human genome is fully identified, and look at this, we have Dr. Askari back with us.
Thank you for having me back.
Okay, great. All right, where we were leaving off is we were talking about the fact that the first patient has been infused with gene therapy, which is very exciting. And the next question I was going to go on to is just what the actual risks are. We did talk about some of them, but are there any other risks that patients or family members should consider as they're considering involvement?
Well, I think the biggest risk is that the gene therapy may not work. We know that from other trials that it doesn't work for everybody. So I don't necessarily want to, overplay that. It's certainly possible that someone can get the gene therapy and it could not work. But that's in my opinion, probably the most likely thing to consider. And then I think it'll work for the majority of people, but obviously we don't know that till we do this. And then the possibility that you could have some mild elevations in liver enzymes is anticipated. So we're talking about liver enzyme elevations of a couple hundred points of value, from like 50 up to 200 or 300 that come down. And one of the reasons that's happening is because of immune responses to the vector. And that's why both these protocols involve some mild to moderate immunosuppression with steroids and sirolimus. So I think those are the main risks that we would think about if we're candidly discussing the risks. Obviously, I think the risks are small, and then the benefits are potentially quite large. That people can come off treatment. We think the gene therapy is going to last, 5 years or more. We're not sure exactly how long it'll last, but that's one of the questions we've been seeing in a chat: is how long do we think this will last? We think it'll last probably 5 years or more in many patients. There's a lot of monitoring that goes on to see, how well the gene therapy is working and how long it's lasting. And so that monitoring is part of the protocols. And so someone has to be willing to do a lot of monitoring to be in the protocols, because it's a lot of safety monitoring. Obviously, down the road, once we know more about how long the gene therapy lasts and how effective it lasts, if it's effective for 100% of patients, some of those things may change the amount of monitoring. But in these studies, there's a lot of monitoring going on. So I think that's the information about safety and monitoring and the length of the duration of the treatment. So there may be repeat dosing in a number of years with another infusion, perhaps of a different vector at that time, slightly different vector. But those are the thoughts.
All right, and that's great, because you've segued into the benefits, which of course we're all very interested in. And you mentioned that potentially the benefits could last 5 years, perhaps longer. I think it's important to differentiate that gene therapy is not the same as gene editing. This is not a cure.
Right. It's exciting to talk about curing Wilson disease. And so, the fact that you're even bringing up the word cure, I think, is an exciting thought. Right now, we know that liver transplant's a very crude form of gene therapy, where you're literally taking the whole liver that has somebody else's genes in it and putting it into someone. And that, quote, cures or reverses the phenotype of Wilson disease. People don't have to take copper reduction therapy anymore. They do have to take a lot of other medications for the rest of their life. And I'm not suggesting that Wilson disease should be treated that way if you don't need a liver transplant. But the truth is that we know that putting these genes in works to make this disease corrected phenotypically, where you don't have to take pills to control copper in the setting of a liver transplant. And we're hoping the same thing will be true with a gene transplant, if you will, with a gene transfer here with these vectors. So while it's not going in and changing your own individual genome, so in other words, just like with the liver transplant, Wilson disease still could be passed on to children after someone's had a liver transplant. Similarly, we would have the same concerns here, and we're not correcting the defect in the germline, nor should we be at this point. So I think we're not talking about a complete cure, but we can talk about a functional cure, if you will, if you're coming off the treatments and coming off copper reduction therapy. And then in terms of a benefit, I think that's one of the big ones, is that, our hope is that people won't have to take medication every day. The copper is also being controlled differently. It's being controlled the way nature intended it, with the ATP 7B gene product or protein. So I think that's another potential benefit. So I think when we're looking at all of these are all reasons why I'm excited about gene therapy for Wilson disease, and why I'm hearing a lot of excitement from many of our patients as well. And even people who don't want to participate because they don't have the time right now are telling me how excited they are this kind of efforts are going on, and that we're trying to get these gene therapies to work for so many patients.
Right, and you've covered somewhat my next question, so I'll just kind of wrap it into one so we can get to some of our audience questions here. Is that we are lucky that we currently have 3 approved therapies for Wilson disease and another that might be approved. So we may wonder, why do we need a gene therapy? And kind of along those lines, for those of us who have met other people with Wilson disease or attended our conferences, we see there is very large variability in this disease and how it affects people. So perhaps if you could kind of talk about all that and how this would help with those challenges.
Well, that's a great question. And the answer is, we actually don't know how much this is going to help. We hope it'll help a lot. But we'll have to see. So patients do have some unmet needs. There are patients who are suffering quite a bit with symptoms. I'm sure there are many on this conference right now. And some of the symptoms may be quite subtle too, that we may not know just how good this can be until it's done, until we see how well the gene correction helps in terms of cognitive problems or thought problems or energy problems or whatnot. Actually, the way we lower copper right now is in many ways very crude. We're giving pills that block copper absorption or that facilitate copper excretion, and copper is not shuttling normally through the body in this. Whereas when the ATP7B gene is in the liver, the ceruloplasmin will be hooking it up with copper normally, the way it does without a defect in the ATP7B gene. And this will be allowing the copper to be delivered normally throughout the body, attached to ceruloplasmin. And if there's too much copper, it'll allow that copper to get excreted from the body, is our hope. And so this is basically, a game changer in how the copper is being lowered. It's not just a fifth way of lowering copper, if you will, that's in there. And of course, many patients can't tolerate those treatments or have side effects from all of the currently available treatments. And so having another option is wonderful for those people. To what degree defects are going to be reversible, we honestly don't know. I mean, there's probably some people with Wilson disease that have permanent damage that even if they get a gene therapy, it won't reverse it. But on the other hand, there may be more reversibility than there is with the other modalities. We won't know until the trials are done. So I don't want to come off as holding out false hope for people. And at the same token, we just don't know how well this is going to work and how effective it's going to be.
Okay. All right. Well, I think everyone's heard enough of my questions. And so Dr. Askari, do you have a couple audience questions there now that you'd like to get to, or do you want me to go with another polling question so you can take a look?
Well, I can try and address one or two of these. I think we've answered the question about one-time treatment or a short period. So it's going to be a one-time infusion in this clinical trial. The trial is set up for a one-time infusion. People are monitored for 5 years in the clinical trial and beyond that as in the clinic. So there'll be lots of monitoring going on, but it's a one-time deal in these clinical trials with an opportunity to extend the trials and come up with a second infusion later on in another trial or in another therapy. So that would be the answer to one of them. And Mary Graper is asking, why are neuro patients not accounted for the trials? And that is a wonderful question, Mary, because at least one of the trials does allow in some people with neurologic problems. So it's not as if these trials are excluding everyone who has neurologic problems. Severe neurologic problems are going to be excluded from both studies, but there are allowing in some people with some neurologic symptoms. So great question, and I'm glad we had a chance to clear that up. Another question is asking me, we mentioned that radioactive copper is used as a tracer. Is this only for the trial to test the results, or is it part of the therapy? Another great question, and I'm glad we can clarify that. This is a tiny dose of radioactivity, kind of like a chest X-ray, and it's used as a tracer or a marker to study how well copper is being metabolized. This radioactivity goes away over a period of hours, really, and within days, the radioactivity should be completely gone. And it's not a part of the therapy. It's a way of measuring this that was developed by our colleagues in Aarhus. So as being incorporated into this study, with some modifications. So I think that's a wonderful question. Do you want me to answer more, or do you want to go to a polling question?
I'll go to a polling question and give you a second to look through all those questions that have come in. So our second polling question is, what Wilson disease treatment are you using at this time? Again, this is anonymous, and just click on the answer that fits you. A is cyprine or trientine. B, zinc. C, penicillamine. D, in a clinical trial with something else. E, none. F, a combination of the above. And G, does not apply to me. So you can see the results coming in here, and it appears that cyprine, the trientine, seems to be the leading medication that those on this webinar are taking at this time, with penicillamine behind, and zinc in third place as far as people taking it. Speaking for myself, I took penicillamine for almost 20 years and did very well on it, and then switched to zinc for maintenance therapy. So we can see there what people are taking. So Dr. Askari, are you ready for some more questions?
Yeah, the questions are great because they're giving me a chance to clear up some things we haven't talked about, or I may not have talked about as clearly as we'd like. So one of the questions is, can the treatment be applied to people at any age, children too? And the answer to that question is, in theory, yes. In theory, this can be given to children as well as adults, but these trials are only open for people 18 and above. So I think that that's an important point. And obviously, I'd expect that once there's more experience in adults, like with many drug developments, they're often developed in adults or tested in adults first and then extended into children. There'll certainly be an opportunity for children to participate in the future. So it's not that they can't be done, but they probably will be done later on. And then the next question I wanted to touch on is, I mentioned sirolimus and prednisone. Are these being taken long-term, lifetime? And absolutely not. These are temporary treatments after the vector is infused. So this is a short-term treatment. And we've just learned, when we were talking about all those other wonderful trials that have been done from the thousands of people and the hundreds of diseases that have been addressed before Wilson disease, we've learned that giving these drugs, prednisone and sirolimus, basically make the gene therapy work better for a greater number of people who get the treatment. So this helps make the treatment effective by toning down the body's immune response to the treatment. And so the trial's written that, the one trial's written that you get prednisone for 58 days. You may be on it slightly longer than that, but you'll be off it in the majority of cases before 2 months are over. So it's a shorter term treatment, not taken for the rest of your life. So that's a great question. In both trials, these are short-term immunosuppressive regimens, not long-term. Someone asked, how do they sign up for the gene therapy trials? You can certainly call my study coordinator or call a study coordinator at another center, and they'll help you go through that. So that's a great way to get enrolled. Another question is asking, are these studies only in the U.S. or they're in other countries? And again, they are being rolled out in other countries. I don't know, to be honest with you, the update on the current status of when those other centers in other countries are open or enrolling. But you can go to clinicaltrials.gov or go to the respective companies, and they'll help you find out other centers, hopefully closer to your home than coming from another country. But there are centers going to be in the U.S., Canada, and in other countries as well in Europe. So I think that should answer your question. Another question is about mutations. Is the therapy applicable to any mutation of the gene? And the quick answer to your question is yes, in the sense that this is not being we're not interacting with your own genes. So you can put in you can supplement any gene mutation that isn't working with this functional ATP7B gene. There may be some study enrollment exclusion criteria depending on whether they can detect gene mutations or compatible gene mutations, but I would handle that on a case-by-case basis. And these companies are willing to check for gene mutations very carefully. So if you might be a candidate and you have Wilson disease and you're not sure what your gene mutations are, or if other people haven't been able to detect multiple mutations, I'd encourage you still to come forward and get screened, because you may well be a candidate for these trials.
All right. Is it all right, Dr. Askari, if I do a third polling question while you scroll through those questions, and I'll be right back with you?
Sure, that'd be great. That'd be great.
All right. Okay. So to find out more about challenges our audience might be facing with medication, here is our third polling question. If you ever skipped your Wilson disease medicine, why did you do so? And the possible answers: A, I forgot; B, I felt good and didn't think I needed it; C, cost; D, drug availability; E, side effects; F, I'm in denial; G, difficult to transport on trips; or H, other reason. And looking at the results, it seems far and away the most common reason is that someone forgets. And as a patient, I can tell you sometimes during the middle of the day, getting that dose in when you're not in a routine or a regular schedule, you can get to the end of the day and realize that you forgot. And it's not that you're being a bad person or anything. It's just kind of that reality of life and trying to remember with everything else going on. So Dr. Askari, are you ready for another question?
Yeah, these questions are really super. So I'll try and go through as many as I can in the time that's left. One is, how is the travel coordinated to the study site if you live in another state? Well, whether you live in another state or the same state, the travel's coordinated through a vendor of the company, and they try and cover the travel costs for you, as well as may provide a small stipend to help you with food and lodging and all those kind of things. So that you shouldn't be losing money having to pay for travel to the sites, and it's all coordinated for you by companies that specialize in doing just that for clinical trials. So, and I think the companies have gone out of their way to try and make this accessible regardless of where you live. About how many visits annually does this commitment time change over the 5-year trial? Well, the two trials have different, requirements, to be quite honest, about visits. But both of them require a lot of safety visits. And many of them require visits that can be done locally as well. There's more visits in the beginning of the study. So obviously the screening takes a couple visits, and then there's dosing and, a few shorter term follow-up visits. So in the first year, it requires a minimum of, I think, 6 or 7 visits. And then there'll be other visits where they come to you and draw blood. So you don't have to actually leave your home or work, but you do have to have a visit by a visiting nurse or somebody just to check on you and be sure things are going all right. And then the visits do continue to occur, but at a lower pace as we go through the 5-year course of the trial. So make no mistake, this does require, I'm not trying to undersell effort being put in on the part of the study subject. This requires a lot of coordination, a lot of willingness to be monitored and be sure you're safe and you're okay and the gene therapy is working. We're also talking, there's a question about, do you stop taking the medication? And in both of these studies, the goal is, yes, to get you off your medication. And so that's requires a lot of monitoring to be sure it's going well, and that the reductions of the standard of care medication, it's done as a dose reduction and a monitoring for how it's tolerated rather than poof, it's gone. So we try and do that in a very safe way and monitor you, but obviously that is going to occur during these trials. Another question is, will this ever be covered by insurance? And I think the answer to that question is, that's the hope of these companies, and that's why they're doing this, is that they hope to get these approved. They've been successful. As I mentioned before, Ultragenyx has done this in it's the 5th disease. They've done it in 4 other diseases. They've been doing this for 10 years or more, and they hope to get this approved to treat Wilson disease. Vivette is working with a co-company, Pfizer, and they're hope to get this approved. And obviously, Pfizer sold many drugs in the world besides this one, and they hope to get this approved and charge insurance companies for it. So I think that's the hope. I think it will be covered by insurance one day. How does it work with patients who get placebo? And that's basically a dose reduction and monitoring part of the study. So they get placebo and they're not off copper control medication throughout the trial. It's a dose reduction and monitoring that goes on for those folks as well. And then they have the option of getting treatment infusion with the vector at the end of 48 weeks. And as I said, in that one trial, people are randomized for every 2 patients who get gene therapy, 1 person gets placebo. And then at the end of 48 weeks, the people who got placebo get the gene therapy.
And Dr. Askari, if you don't mind, I'm going to interrupt you there because we're almost coming up on the end and we have one last polling question to get in. And while we're doing that, if you could think of any parting thoughts you'd like to leave with us or anything that we did not think to ask you that you'd like to share. So going to that last polling question is, did you learn from this presentation? Just 2 answers, yes or no. We will not have our feelings hurt. I see there are no no's, so that's very encouraging. Thank you, everyone. And back to you, Dr. Askari, any parting thoughts?
Well, I just want to thank everyone, Wilson Disease Association, obviously you, Rhonda, and the sponsors. All the work that's been done on these therapies for Wilson disease is very exciting, both for those of us who treat Wilson disease and for the patients and families themselves. And we're obviously very excited to see how these gene therapy trials enroll, and I hope to be able to report some exciting results back to folks in the future about how these gene therapies are working and how people are feeling after they've had them. So I really I want to thank all of you, both the study participants and the people with Wilson disease and the Wilson Disease Association and sponsors for everything they're doing to help make treatments for Wilson disease better for everybody.
All right. Well, thank you. Thanks for thanking everyone. So I don't need to do that. So I am just going to thank you for taking this time out of your Saturday to join us and explain all this to us. And I'd like to let the viewers know, if they want to view this program again or share it, they'll be able to find it on the WDA's website under Genetic Therapies, in the treatment section under the Living with WD tab. And it will also be on our YouTube channel. So once again, thanks, and I hope everyone has a great Saturday.